All right. Sorry that we're running 5-ish minutes late. If you haven't noticed, this is an old building. We have some terrible computer and audio equipment that we're working with at times. But anyway, I'd like to welcome our second seminar speaker for the fall 24 FEC grad seminar series. Our speaker today is Dr. Casey Youngflesh. Dr. Youngflesh is currently an assistant professor in biological sciences here at Clemson University. He has a Bachelor of Science from UC Santa Cruz, a Master in Conservation Biology from New South Wales in Sydney, Australia, and a PhD in Ecology and Evolution from Stony Brook University in New York. Prior to joining Clemson, he has been a NASA graduate fellow and a postdoctoral researcher at institutions such as UCLA and Michigan State. Dr. Youngflesh is a quantitative ecologist who combines ecological theory with cutting-edge quantitative tools to understand the drivers of biodiversity and population dynamics and how these processes vary across scales. While he's interested in a broad range of ecological systems, he primarily uses birds in his research to develop reproducible analytical pipelines that leverage big data approaches such as hierarchical Bayesian models and deep learning. Today, he'll be telling us a bit about characterizing ecological responses to global change in the age of big data. So welcome, Dr. Youngflesh.
Thanks for having me. Thanks, Jared, for the invite. Firstly, I'm just across campus, Long Hall 206. If you ever want to chat, be very interested, drop by or send me an email. There's a lot of, I feel like what I do, a lot of folks do that same thing over here in this department, just a little bit of a different flavor. So I think there's a ton of overlap in ecology, and there should be no reason why our two departments, I don't know, can't interact more in those areas. So really happy to be over here. Let's see if this works. All right. So understanding how ecological systems are responding to global change stands as one of the greatest challenges that we currently face in the natural sciences. Given the complexity of these systems, this of course is this inherently challenging task. Much of what we know about these systems is limited in scope, limited in spatial scale, limited in taxonomic scale, but really forced to generalize our findings quite widely. We want to understand these systems. And really developing a holistic understanding of our natural world is going to require us to characterize interactions across the entirety of ecological parameter space. That is, we need to explore the variation that exists across these spatial, temporal, taxonomic, and probably many other scales. In this age that we're currently in, of course, we have this access to an incredible array of data resources, both new data streams that are just now coming online, as well as existing long-term data sets that we're now using in different ways through the use of newly developed quantitative tools and computational resources. This is allowing us to do things, explore new questions, and to approach existing questions in new ways. The age of big data, it's not just about the data themselves. It's about these compute resources. It's about these quantitative tools, things like hierarchical Bayesian models, things like machine learning, to actually make use of these data streams. And these tools, as well as the data, have advanced tremendously over the last several decades. So as a researcher, I operate at the intersection of ecology and data science. Just move that mouse out of the way. Using a lot of eco-informatic tools, I would say, to explore both basic and applied questions of ecology. So I'm interested in developing these frameworks that leverage a variety of data resources to characterize interactions, mostly at the population community level, how things vary across these different axes of variation, and ultimately to address how ecosystems are responding to global change. So again, as Jared mentioned, much of what I do is focus on birds. And if you realize that there are an incredible array of data resources for birds, it's one of the reasons why I work on birds. They're also quite fascinating. But really the taxonomic interests are, it's kind of taxonomic agnostic, I would say, many of my interests. So today I'm going to talk to you about three areas that I and now my lab has been working in. Firstly, phenology and biodiversity, and then talk a bit about population dynamics. Provide a few examples of how we're approaching this work with particular focus on how we might think about these things in the context of global change. So firstly, phenology, that is the timing of seasonal events. events. While often underappreciated, phenology plays this critical role in ecosystem functionality. So organisms need to time important life history events to coincide with events at other trophic levels. So if you're a bird, you probably want to breed during a period where food is readily available, so insect emergence in that spring. And if you're an insect, you need to time that emergence in the spring to coincide with the availability of vegetation. So greening up in the spring, those nice powerful leaves if you're a caterpillar. And here I'm framing the importance of phenology in terms of these trophic interactions, cross-trophic interactions, though it's certainly important for other reasons as well. Concerningly though, in response to climate change, we're now seeing a shift in when these phenological events are occurring. So in general, spring phenology events are occurring earlier than they were in the past. So in aggregate, we know birds are migrating earlier than they used to, for instance. And these changes might lead to some phenological asynchronous, where birds, for instance, are breeding outside of some optimal window of time. Yet while we have all of this evidence that phenology is changing, much less is known regarding how and why these responses vary across space and across species. So I was interested in exploring the variability in phenology, how it's responding to global change for a large number of North American bird species, specifically this area in eastern North America. So for these birds in particular, phenological change is potentially concerning given the large-scale declines we've seen in many North American bird species. So a 30% decline, about 3 billion birds lost in the last 65 years, according to recent estimates. Now, of course, working at these large spatial temporal taxonomic scales, it's challenging, right? It comes with these data requirements. Well, what are we to do? Well, luckily, as ornithologists, we can leverage the power of our communities. We can leverage the power of birders, bird enthusiasts, to go out and collect data for us. So if you're not familiar with eBird, this is one such platform that people use. It's an online tool. You essentially go and report where and when you've seen particular species of birds. So for this red cell, there will be a number of checklists, eBird checklists collected by birders throughout the year. So in a given year, in a given area, a number of checklists. And in some of those checklists, a target species, say red-eyed vireo, is recorded, so a record of presence. In other checklists, that species is not recorded, so that's a record of absence. You can use these presence absence data to derive estimates of when birds are arriving into these cells for each year. So circles at the top of the plot represent checklists where red-eyed vireo was recorded in this particular area for a single year. And checklists at the bottom here represent checklists where red-eyed vireo was not recorded. So we have day of year on the x-axis. In black, we have the model fit for logistic GAM, where that equation is saying the 0-1 data, presence absence data follows a Bernoulli distribution, where that probability varies as a smooth function of day. So that smooth function is what characterizes this generalized additive model, that GAM model. It's fitting a wiggly line to that data. So to interpret this year, we give us a very low probability that you see red-eyed vireo in a checklist in this particular cell, probably eastern North Carolina somewhere. Then that probability rapidly increases during spring, as we'd expect. So migratory species, they're coming up. You're more likely to see red-eyed vireo in these checklists during that spring time. So we define the point at which this curve is half of its maximum value. We call this the half max. And this is our metric of bird arrival phenology. We can quantify some uncertainty about that as well. That's shown in these dashed lines right here. So I ran these GAMs for each species, cell, year, for about 56 different species, 16 years, amounting to about 7 million bird observations, deriving these estimates of bird arrival. So these are some spatial maps essentially showing the results from those GAM models. Each map is a different species year. So blue hues are later arrival, pink hues are earlier arrival. As you expect, these birds are migrating south to north during the spring, so it's what we expect. So this is all done in a Bayesian framework, so we're propagating that uncertainty throughout the entire analysis. We're quantifying the uncertainty in that curve, quantifying the uncertainty in that half max, and piping that through to make sure we're not losing anything in the mix here. So what we've done is essentially distilled this information that we've crowdsourced from the community, from birders, into this phenological data product now. And I think it's really worth noting that this wasn't something that was even computationally tractable even a short time ago. if you were to run this on your laptop as opposed to high performance computing resources, you'd probably be waiting a couple of years for it to finish. So using this data product, I wanted to understand how birds are responding to phenological changes in terms of global change. So for a given cell, for a given species, we'll focus on tree swallow here. We have information on when birds are arriving, that's shown in blue, and when plants are greening up. That is really our metric of spring arrival as measured by satellite-based sensors. Literally the greenness on those trees as it ramps up. So we have year on the x-axis, phenological anomaly on the y-axis. You can see those things somewhat co-vary. They vary through time, but that's really what we're interested in, right? Then using a spatially explicit hierarchical model, I wanted to characterize the sensitivity of birds to these changes in green-up. Where sensitivity is defined as for every 1 day change in green-up, how much are birds changing their arrival timing? So a sensitivity value of 1 would represent perfect tracking. 1 day change in green-up, 1 day change in the migration timing. So the green hues here represent higher sensitivity. These hay, straw-colored hues represent lower sensitivity. So what we can see is that there is, for this species at least, this clear latitudinal gradient in how sensitive birds are across their range to changes in green-up. And this is the pattern we really see across most of the species, this higher sensitivity at higher latitudes. And this essentially tells us that birds have this capacity to adjust green-up as they're moving forward during their migration. So if this species is migrating across the Gulf of Mexico, they might not have a very good idea about what's going on down here, if they're in the Yucatan or something like that. But as they move up, they're adjusting, they're determining how long to sort of rest and how fast to fly, so to speak, from a population level to get up to where they want to be during the breeding season. So we also found substantial differences among species, as you might expect. So for some species, prairie warbler, very low sensitivity, very small gradient across latitude compared to something like tree swallow, that's just much more sensitive, has a much stronger gradient. And there is some variety among species. Actually, northern parula here, you actually see the opposite pattern. And there are actually some explanations for that having to do with migration corridors, we think. But in general, most species kind of do what tree swallow is doing here. But I would say importantly, these differences are predictable according to species level traits. So species that migrate more slowly, arrive earlier, and overwinter further north tend to be those that are tracking green-up, the arrival of spring, more readily. So we have arrival sensitivity on the y-axis, each point is a species here. And this x-axis is a composite score. It's the first principal component for these 3 species traits. These 3 things co-vary quite a lot. So if you're a slow migrator, you also arrive early and overwinter further north. So we can't really decouple those things. So this relationship, I think, makes sense because if you're a bird that overwinters quite far north, you probably have pretty good information about what's going on in the breeding ground. There's spatial autocorrelation in environmental conditions. So the closer you are to somewhere, the better information you're going to have about what's going on there, as opposed to migrating 1,000 kilometers or something where you really don't have that great of information. And I think this can really give us a sense of which species have a greater capacity to adjust to future global change, to future phenological change, which species might be more sensitive. So this is all for sensitivity of arrival when birds are migrating, when they're showing up on their migratory breeding grounds. In some other work, I found it's very much the same story if we then consider when birds are actually breeding, which is kind of a different thing, maybe a little bit more important, you could argue. But in both cases, for both arrival and breeding, it's clear that birds are not keeping up with climate change. They're not keeping up with this phenological change of spring. So here we have bird phenology on the y-axis, green-up advance on the x-axis. If birds were keeping pace, that would be represented by this dash 1-to-1 line here. But instead, bird breeding would need to be about 3 times as sensitive as it is to be keeping up with climate change. Arrival would have to be about 5 times more sensitive. So they're lagging quite a ways behind, I would say, which is potentially concerning. So over time, as green-up is changing more rapidly than arrival, we might get this temporal decoupling that's happening, that's occurring. So this blue band here represents peak resource availability, when the resources are available to these birds. The red line Well, when these birds really need these resources, we have day of year on the Y, year on the X. At the beginning of this period, these two bands overlap, right? You've got the resources when you need the resources. We might call that a phenological match. But over time, as the phenology at these two trophic levels is diverging, changing at different rates, these windows no longer overlap. So this temporal decoupling happens with the potential for some phenological mismatch to occur, where I'm really describing mismatch as some phenological asynchrony that has the potential for demographic consequences. So something for the demography of that population. But we have overwhelming evidence now that phenology is changing across a number of different taxonomic groups all across the world. But we really don't know much about the ecological consequences of this asynchrony. We have these theoretical expectations, we've talked about them a lot, but we really don't know how this plays out in nature on these large scales. So in order to address this question, I used data from a large-scale bird banding project known as MAPS by the Institute for Bird Populations. This is bird banding done during the breeding season, so spring and summer months here in North America. All of these points are MAPS banding stations. These are just stations located in forested environments. So those are study species of interest. About 41 different species, mostly songbirds here, something that you catch in a mist net. It's all standardized bird banding, I should say, as well, standardized effort. Every location is doing the same sort of protocol. And then from these banding records, we can calculate an index of breeding productivity. So how many young that are produced for specific species, for a specific station, or a specific year, as well as an index of phenology. We broke phenology down into two components here. Firstly, we have an early late index, where positive values are years where both green up and bird breeding are late in the year. Negative values where both of these are earlier in the year, and zero values represent sort of an average year. So again, this is a composite score coming from a principal component analysis. The second index is this asynchrony index, where positive values represent earlier green up, later breeding, negative values, the opposite, and zero values represent average synchrony among these two phenological events. I then model breeding productivity as a function of these two indices for each species, station, year. So here we have the results for this model for a single species, just to show the pattern here. Song sparrow, breeding productivity on the y-axis. On the left, we have our early late index. On the right, we have our asynchrony index. What we can see is that there are values for each of these indices for which breeding productivity is maximized, right? There's this hump-shaped curve. So you're producing fewer young if you breed too early or too late, or if these two phenological events are out of sync. So here are the results for all 41 species. Each line is a species. These are all modeled together hierarchically. And we can see this characteristic hump-shaped pattern, this concave down pattern that we expected to see. So visualizing this a little bit different, same thing in a different way. This is the cross-species effect. So we have the asynchrony index on the y-axis, the early late index on the x-axis. This yellow area is this area of phenological parameter space where breeding productivity is maximized. So high breeding productivity in yellow, lower breeding productivity in blues and purples. So that productivity is falling away from this peak. And this is really what we expect, right? This is really what we hypothesize, that there would be this Goldilocks area for phenology. So it's pretty exciting to actually see this come out of the data. So the phenological dynamics explain about 5% of the variation in breeding productivity, despite the many factors that we know are going to affect how many young birds produced. We see this population, we see this signal at the population level, really across a wide range of species and a very large spatial extent. Essentially, the whole US, a little bit of Canada, I would say. I would say this stands as some of the largest scale evidence of the importance of phenology for demographics in any taxonomic system. So really returning to our earlier question, is phenology, is phenological change, phenological asynchrony important for the dynamics of this system? I think the answer is clearly yes for these species. So using these estimated responses, along with projected changes in the arrival of spring, we might expect about a 12% decline in how many young birds are producing by the end of the century. So on the Y, we have percent change in breeding productivity. On the X, we have green up advancement. This 25 days, that's a likely scenario by 21, by the end of the century, for how much earlier spring is going to arrive in some of these forested systems. So really, if these species are unable to adapt or somehow cope with these phenological changes, this might present an additional risk to the persistence of these populations, which are already clearly under threat from habitat loss and a number of other factors. So while I've spoken to you mostly about the link between birds and greenup, there's really this missing trophic level here, right? There's insects, what birds are actually eating. These birds are feeding their young, mostly larval moths, so moth caterpillars, not too many butterflies, mostly moths. So we've been trying to integrate this into this tritrophic framework. And I will say that the kind of short of it is that insects are really hard, right? Really diverse in North America, at least the ones that birds are eating. Many of them are multi-bolting, so multiple generations in a single season. We don't have that great of data on lepidopterans compared to birds at least, so I think we're kind of spoiled as ornithologists for data. Certainly a data mismatch there. But we've been doing what we can, despite these challenges. We've really been trying to look across these three trophic levels to try to understand how these different trophic levels are responding at different or similar rates to global change. So this is some synthesis work being led by Michael Belitz, who's now a postdoc at Michigan State with Louise Zipkin. Basically what we're doing here is looking at the effect of growing degree days, this temperature accumulation in the spring. How is temperature impacting when these different phenological events are occurring? We have green up, so plants, how they're responding to temperature, how leps are responding to temperature, and how birds are responding to temperature. And what we see is that essentially leps and plants are responding at similar rates to changes in temperature, but birds are much less sensitive. So if we're thinking about this in this cross-trophic, these two different links here, it's really this bird-lep link that we should be much more concerned about as opposed to this lep-plant link. I also think this provides nice justification for using green up as our sort of proxy for food availability, phenological food availability for these birds. So in order to increase the accessibility of the research, the data, my collaborators and I built this online interactive web app using RShiny. If you haven't used RShiny before and you are passing data to managers or people like that, I think it's a really great tool, check it out. So our goal with this really was to provide a platform where folks can go and interact with the data, download it, explore results for themselves, which I think is pretty cool. And then following on from that, my colleague and I, we developed this teaching case study focused on this work. So if you're looking for an advanced undergraduate level or graduate sort of lesson plan focused on phenology, whether that's just questions and whatnot focused on this paper, or if it's something you're looking for, a data analysis class and you want a cool data set, that's all built into this lesson plan too. Get in touch, I'm happy to provide additional information about any of that stuff. Yeah. So I've also been exploring the impact of global change on other dimensions of ecology as well, including biodiversity. So in this case, morphological variation in bird populations. So morphology is both a cause as well as a consequence of how organisms interact with their environments. Much of the morphological literature is focused on differences among species, which of course can be quite large. Cardinal is very different from a condor. Substantial differences also exist within species. And I think this intraspecific component of variation, of phenotypic variation, is really an important element of biodiversity that we often forget. We're often thinking about biodiversity, species richness, but it's really across all of these levels of biological organization. Of course, abiotic, biotic conditions that might literally shape species, those are going to vary across a given species range. The temperature, for example, shows substantial variation across latitude. Areas near the poles are generally colder than areas in the tropics. And given the importance of body size for thermal regulation, we might therefore expect animals to generally be larger at higher latitudes. So this idea was first proposed by Carl Bergman many years ago. generally known as Bergman's rule. So for large animals, you have a smaller surface area to volume ratio, less likely to lose heat due to convection compared to a smaller animal. So these proposed associations between temperature and body size over space have motivated expectations for how we think endotherms are declining, how endotherms are responding to these warming temperatures, I suppose. It's been proposed that animals are generally declining in size to cope with these thermal stresses. However, the empirical evidence here hasn't always matched our expectations, and these temperature body size associations, even over space, have been argued about continuously in the literature for the 175 years since Bergman's rule was first proposed. So here I wanted to leverage large-scale data, again from the MAPS program, to really understand this variation over both spatial as well as temporal and taxonomic scales. So this is the full extent of the MAPS data set project, I guess, about 1,100 banding stations distributed across North America, really to answer the question, how are abiotic conditions shaping avian morphology across North America over the last 3 decades or so. So this amounted to about 250,000 bird captures, a little over 100 species. And again, mostly songbirds here, things you'd be catching in mist nets. So all these bird banding projects, they're typically implemented to do some demographic analyses. Morphological measurements are often taken as well when you capture these birds. So measures like wing length, measures like mass. And we could use these to create some indices to assess the morphological variation across these axes. So there's some covariation between these traits. Of course, if you're a bigger bird, you have longer wings, you're probably going to be a heavier bird too. So first step here was to derive 2 morphological indices where the size index says, all right, how large of a bird are you, a big bird or small bird? And the y-axis here, the wing index, that's how wingy are you, so relative wing length. So for each individual bird, there is an estimate of a size index and a wing index. I won't go into the whole derivation, but I'm happy to talk to folks about it afterwards if you're interested. So we then quantified how these 2 indices, the wing index and the size index, vary across space and time, as well as in response to temperature. So I found that within species, birds are typically larger at higher latitudes. So the gray lines here, each one is a species. The black bold line is the cross-species estimate. They're all modeled together. And these results are in line with Bergman's rule, matches our expectations given what we know about thermal tolerances, since higher latitudes are generally colder environments. I also found that birds are generally declining in size over time, as we might expect, given that temperatures have been warming over the last several decades. But while these things match our expectations, is this really temperature that are driving these patterns? It could be a number of different things. So I found that warmer temperatures are associated with smaller birds over both space and time. So here I'm showing you the effect of really temporal variation in temperatures on a single site. It's the cross-site, cross-species estimate here. And this relationship holds true of both temperature in the year of capture, as well as 1 year prior to capture. So these birds don't live very long, they're breeding in that first year. So the fact that this effect persists across these multiple years suggests that it's not just something due to morphological plasticity, developmental plasticity, but there's likely some size-dependent mortality that's going on as well, depending on temperature here. So interestingly, if we take this one step further, even within species, we see differences in the response to temperature. So the white lines are isoclines here across the range of the red-eyed vireo. So it's one of 100-odd species we analyzed. Red hues represent that temperature is having a larger effect on body size over time compared to these more yellow hues. So you can think of the color here as literally the slope of this line, the slope of the association between the size index and the temperature anomaly. So what do we see? We see a clustering of these deep red hues in the warmest portions of the species range here in the American South here. So this tells us that it is likely the warmest rather than the coldest temperatures that are driving these body size temperature associations. It also provides widespread support for Bergman's rule over both space and time. So warmer temperatures mean smaller birds, and that's true whether we're comparing South Carolina and Maine, or whether we're comparing the year 1990 and 2020. So I think one real critical finding here is the fact that the spatial response to temperature is much larger than the temporal response to temperature. It's about twice as large, that spatial response. So while the fact that, well, birds are changing in response to temperature, that might tell us that there's some adaptation or some, I don't know, plasticity there, that birds are coping with this change. This point suggests that they're probably not changing fast enough. And given that temperatures are projected to continue to increase, we do expect to see these trends continue into the future, though with somewhat unknown consequences. That's other abiotic, biotic factors no doubt play an important role in this as well, shaping morphology, though have received less attention. So things like air density varies across an elevational gradient, generally lower air density at higher elevations, right? We might expect larger wings, proportionally longer wings, to cope with that reduction in air density. So there are literally fewer molecules of air at higher elevations that reduce the amount of lift that a wing provides. That's what makes it more difficult to breathe at higher elevations. So certainly important in terms of climate change as well, given that, well, many species are being pushed upslope, right, as temperatures warm. And starting this work, though, a number of people have said to me, well, surely these differences in air density are not actually important for these birds. Well, I think the great thing about air density is, unlike many things in ecology, we can actually derive that from physical equations. There's no noise here, people. Temperature on the y-axis, elevation on the x-axis. We see that you generally have lower air density at higher elevations, and it's generally less dense at higher temperatures. So less dense yellow, more dense purple. And we can map that onto the North American continent. You can see a clear effect of elevation through the Appalachians and certainly through the Rocky Mountains. There's a big elevational gradient there. And while these units, kilograms per meters cubed, probably don't mean much to you, I think the proportional difference is important. So almost double the air density at the top compared to the bottom here. It's pretty dramatic. So for every 1,000 meters increase in elevation, you get a 10% reduction in air density. And that's taking into account temperature as well. Now, the lift that a wing provides, that is going to be proportional to air density. So that means a 10% reduction in air density is a 10% reduction in the amount of lift that a wing provides. Now, some of the species I looked at range over 3,000 meters across their elevational range. So if we took Wilson's warbler, for instance, if you took an individual Wilson's warbler from the bottom of its elevational range, brought it to the top of its elevational range, its wings are produce 30% lift, less lift in that environment. It's pretty dramatic. But what do the data say? Do we see this morphological variation across these elevational gradients? Well, within the species, we do see that. So again, each line is a species here, larger wings at higher elevations, even within species. Now, the notion that birds have larger wings at higher elevations is not new. However, wing length was always taken to be an indicator of body size. People thought that birds were just larger at higher elevations because it was colder there. We show that is actually not the case. Birds are, if anything, a little bit smaller at higher elevations. They just have longer wings to help them, we hypothesize, cope with that reduction in air density. So this is the first large-scale cross-taxonomic demonstration really of this pattern, this universal increase in wing length, slight decrease in body size with elevation. So together, I think these findings really illustrate how there are these non-mutually exclusive processes that are all acting in concert to shape the morphology of species. And I think there really exists a gap in our understanding of what's driving these morphological processes. And I think that has some really important ecoevolutionary implications in understanding how these species are responding to global change. And there's certainly a lot more to really unpack here. So one of the areas of research that my lab is really focusing on right now, this is work being led by Viviane Zulian, postdoc in the lab, this idea of cross-scale biodiversity. So we can think of variation within the population level for a given species, across populations for a species, across communities. So really increasing levels of biological organization as we move to the right here. So this is some real preliminary work that we're doing, but as one example, we have variation within populations, how much phenotypic variation we have within populations on the Y for body mass and wing length, that actually declines as we move to more northern latitudes. So this is just one of the mechanisms that we're looking at, but really the mechanism behind this, the thought behind this is that, you know, most of the North American continent was covered by a glacier during the last glacial maximum. And as that glacier retreated, these birds had to colonize or recolonize these areas further north, potentially through a serial founder effect. So you're essentially losing morphological variation as you move northward. You're losing that, you're losing that variation within populations. So I guess to say more to come here, preliminary results. I think there's some really cool mechanisms behind this, different patterns that might drive this phenotypic variation. And again, I think a lot of important implications for understanding which populations might be more robust or more adaptable to changes into the future when we have that additional phenotypic variation. So I told you a bit about global change in the context of ecology, biodiversity. We also work on questions related to population dynamics. I won't go into a lot of detail here, but I do want to highlight some of the cool ways that we can use, especially some of these new data streams that are coming online, to understand biotic-abiotic relationships and how they're related to demographic processes. So this was part of my dissertation work. We really wanted to understand what the drivers of Gen 2 penguin demographics were. So this is me in the field setting up a camera at one of these penguin colonies. You won't see me in the field much these days, but it is proof that I have spent some time in the field in the past. This is all built off the Penguin Watch Network. This is run by a collaborator of mine, Tom Hart, in the UK, where essentially the idea is to install these robotics cameras, which I'm sure many of you are familiar with, set to collect time-lapse imagery at these penguin colonies, really providing this high-resolution window into what's going on here. We wanted to know what's driving demographics, whether it's human disturbance, krill fishing in the region, or other abiotic processes. So this is an example image of a colony. The red dots are chicks that we've annotated on those images. The green lines are a tessellation that essentially delineates nest zones for modeling purposes. So we had a number of these images at each site where image was collected every hour, I believe. So using nearly 10,000 of these images, we, I guess from about a dozen colonies across the region, developed a hierarchical model, sort of modified marker capture model to estimate the impact of these different things on penguin demography while accounting for this observation problem. So this is a little schematic here. If we go through time to the right, each of these rows is a nest. Sometimes you see the chicks, sometimes you don't see the chicks as in a marker capture framework, but you don't know if it's because the chick is hiding behind the adult or because it's truly dead. So we're probabilistically trying to determine what's going on, get this latent information, so to speak, from this data. So I think one really interesting takeaway, and I think my favorite takeaway from this work, is the importance of extreme weather events for breeding productivity. So this is a time-lapse video of a penguin colony. And on the right is the estimated number of chicks in this image. If you look really carefully, you can probably see some chicks on the adults here. You'll see right when this large snow event happens, we see a sharp reduction in the number of chicks. So essentially what's happening here is this large snow event comes in, it wets out all the downy chicks, and they literally freeze to death. For those of you that have been caught out in a rainstorm with a down jacket, you can attest that it doesn't really provide much insulation when it gets wet. And so these penguins, they're penguins, but they are literally freezing to death because they don't have that adult waterproof plumage yet. So I think this really illustrates how we need these really high temporal resolution data sometimes to understand these interactions between the abiotic and biotic environment. Yes, big data can mean large spatial scales and whatnot, but we also need high temporal resolution data. And I think with this increase in extreme events, which are by definition hard to detect, hard to gather data on, you really should be moving towards thinking about these high temporal resolution data as well. So another data stream that I'm particularly interested in is satellite imagery. So collecting data continuously across the globe. We've been using satellites to monitor abiotic conditions, vegetation for a really long time now, decades and decades. But I've been exploring how to use this to actually monitor animal populations directly as well. So for instance, we're currently working on a project, developing a project focused on Pacific walrus, really looking to understand how populations are responding to global change in the North Pacific. So walrus, important benthic predator in Alaska, in the North Pacific, and a species of really substantial cultural and economic importance to many indigenous coastal communities in both Alaska and Russia. And we really don't have an idea how many walrus there even are, or how they're responding to these changing conditions, disappearing sea ice, increasing fishing pressure. As you can imagine, studying walrus is a difficult task in the field. This is not a hospitable area to do range-wide surveys in. It's very expensive. It's actually quite dangerous to be flying planes or helicopters around this region. Technological advances in satellites, though, are fundamentally changing the opportunities here. Walrus are very large animals, of course. Each one weighs about as much as a Volkswagen Beetle. We're also incredibly gregarious. So this blob here, it's all walrus. It's about 10,000 walrus all lumped together, touching one another in these giant terrestrial haulouts that kind of haul out of the water. So that image is from a plane. You can imagine when you have so many walrus, you can also see that from satellites. So the image in the middle is from Sentinel-2 satellite. As you guessed it, the brown blob is walrus. The blue here is ocean. This is a barrier island. It's like a sandbar. And the pink is all vegetation. So that's a false color image, we call that. That's the near infrared band that's showing up there. So, okay, we can see it, but what we really want to do is automate this process, right? We want to train a computer to find the walrus for us in this image. And we do that through the use of convolutional neural networks, CNNs. So deep learning model used for a bunch of different things these days, facial recognition, medical imaging. We're essentially training a computer to do this by telling it what is and is not walrus. So for each image, we can go in and annotate it and say, okay, well, black areas are walrus, white areas are not walrus. And that model learns. It learns what it looks like, what characteristics are associated with walrus. So this is some preliminary work that we've done. This is the satellite image. There's a walrus haul out on the end of the spit here. This is my annotation, what I said was walrus, black walrus, white not walrus. And this is the CNN's prediction of what it thought walrus was in that image. Now, importantly, this image was not used to train the model. So this was held out, we trained the model with some other data, and then we tested that on this to see how well it does. So the darker pixels are higher probability of walrus, lighter pixels, lower probability of walrus. You can't really even see the gradation here because it does pretty well, which is good news here. So it already works pretty well, pretty precisely, and pretty accurately. And what's really exciting here is that we're now getting these images across the global range of the species, about 2 million square kilometers, with passes every couple days at these high latitudes because satellites, the orbital paths overlap quite a lot near the poles, which is good news for us. So we intend to apply this to the existing catalog of satellite imagery, dump this information into a database, and really understand how walrus are responding to changing conditions. Beyond that, really from a management perspective, we can then take all this information and every day say, all right, what satellite imagery has been collected the last day, download it, classify it, dump it into the database, and ship that information to stakeholders through something like a web-based management tool, something like a Shiny app or an email or whatever it is for people that they want to, whatever information they want. And this will now allow them to set exclusion areas around walrus haulouts at particular areas in time. For instance, in addition to the sort of big picture perspective of, all right, how many walrus are there? Should we be considering them under the Endangered Species Act, things of that nature. So working with folks at the USGS Walrus Program in Alaska, and as well as the Eskimo Walrus Commission, which is a consortium of indigenous walrus hunting communities in the region that actually co-manage populations with U.S. Fish and Wildlife Service to really figure out, all right, what do they want? What do they need most from this sort of framework? And I think these types of tools, they're really only become more useful and more common as the availability of these types of data continues to increase in the future. And this, of course, is not true of just satellites. It's true of a number of different data streams that I know folks in this room use a lot. Everything from community science to camera traps, UAVs. There's been an explosion of these data resources recently, and it's really not going to slow down in the future. And I think, again, the key to taking advantage of these things are the advances in compute resources resources, quantitative tools to actually make use of these data, to harness the data revolution, so to speak. Flexible approaches that integrate the uncertainties and all the complexities that these big data sets have to really understand what's going on. I think this is what really excites me as an ecologist. So using these approaches, we can ascertain all sorts of cool things. I think about natural environments. I told you about how we use this to understand how species are keeping pace or not keeping pace with climate change. Told you about this in terms of phenology as well as morphology. Told you about how these responses often vary predictably across species according to species level traits. And I showed you that based on projected phenological increases, well, it might not be looking so good for birds, given on how well they are responding to climate change and phenological change so far. So I think one of the challenges to understanding global change moving forward is to integrate our efforts across our disciplines, different subfields, different languages, different data sets telling us different things. But as we build up to this integration, I think these sorts of quantitative tools, approaches, have a really important role to play in developing that community and trying to bridge these disciplines. So I'd like to acknowledge funding sources. Some of this phenology work was funded on an NSF grant that I was a postdoc on. Thanks to Michigan State University for a postdoc fellowship funding. Many folks for the compute resources to make this possible, my collaborators. Big thanks to all the folks that contribute to these community science projects like BIRD. This type of work would not be possible without the hard work of so many different people. That is the age that we live in. There are so many amateur scientists contributing to our work to do this cool stuff. So big thanks to all those folks as well. Yeah, finally, thank you very much for listening. Apologize for going a little bit over, but yeah, thanks for the opportunity and happy to chat now or later and answer any questions.
So I've worked with some deep learning models and some convolution CNNs, and two questions about that. One, just curious, how many samples did you have to use to train that CNN of walruses?
Yeah, good question. So you have the Sentinel-2 images that come in like a larger tile, and because of just the computational constraints, we broke it up into a number of different tiles. I'm thinking the number of larger images that we actually annotated walrus was in the hundreds. So it wasn't that much as far as deep learning goes. You don't need 10,000 samples or something. And I think that's largely because it's just so obvious in the image, right? You've got this brown blob on something that is very distinct from that. But it presents a challenge because from a class imbalance perspective, where there's lots of non-walrus class, but there's not that many walrus class. So thinking about how to balance that in the training of the model, I forget what the technical terminology, but you can kind of set it up to kind of taper off the number of walrus that it's seeing throughout the training to help it train a little bit faster. But yeah, low numbers of, I would say, annotated data compared to many projects, but it was still a lot of work just to annotate that stuff.
And do you see that maybe changing with like planet satellites getting a lot better and we're getting centimeter data, maybe using that to monitor bird migrations?
So planet data, I think, has there's a huge opportunity there. So planet data, we're collecting 3-meter resolution data, 3 meter by 3 meter, as opposed to 15 meter by 15 meter in Sentinel-2. I think that's great. Two things about that though. One, planet data is commercial. You can get access to it, but it's hard. And when are they going to pull that? It's not publicly available stuff. The reliability of the sensors aren't quite as good as something like Sentinel. So depending on your task, maybe there's something there. But I mean, yeah, if not planet, something else. Like we're only getting better. We're only getting better spectrally and spatially to poor resolution. So I think there's huge opportunity, and planet is part of that for sure.
With the CNNs, and forgive me if this is a dumb question, but with the CNNs, after you trained them to detect the walrus, what was like the time difference between them calculating what they thought were walrus and you manually indicating what the walrus were, if any?
Yeah, like infinite, I don't know. It's real quick
Okay.
to classify an image, and to go in. So this is like semantic segmentation, we'll say, or it's like pixel-wise identification. So that's another thing about the replication where like, if you're training a CNN to recognize cat or puppy or something, each one of those images is like one, like an N of one. But for an image of walrus, they're not like completely independent, but you might have like 1,000, like 100 pixels there or something like that. So yeah, it's pretty quick. So we figured out, in thinking about, can we actually scale this range wide if you have 2 million square kilometers? How much time would it take on, a high performance GPU machine on, Palmetto 2 or something, or AWS? And, I think it's something like classify a year of data in about a day or something. So it's like not bad at all. Yeah, it would be very impractical, I think, to go through as a human and do it. But I think it brings up a good point where, sometimes the CNN performs better than others, and you could set it up in such a way where, all right, you want to flag all images that could possibly be walrus, even if they're very low probability, and then have people go in and check a subset of those images. So it would still save you a ton of time, even if it's not 100% perfect. So yeah, good question.
Casey, there seemed to be, going back to your isoplet in your first part of your talk with phenology and looking at bird nest success versus asynchrony and phenological change. It seemed like, I know that was for all species, but that high area of high reproductive success seemed like it was offset just a little bit with the asynchrony, and I was just curious if you provide some offset, some insight into what was going on there.
Yeah, I totally agree. It's very curious and interesting that, yeah, birds don't seem to be perfectly matched up to where we might expect them to be. And I think that is partially due to the fact that there are basically larger consequences for being on one side compared to the other. So it's better to be a little bit late than to be a little bit too early. Because you show up too early, well, it's going to be winter or something. You show up a little bit late, well, maybe part of the buffet is already gone. So I think that's probably one of the reasons we're there. Although related to that, this is kind of quite interesting. As green up changes, bird productivity, we do expect to actually increase a little bit from solely a phenological perspective. So yeah, they're not at that optimum. They're going to be moving over that optimum and then across that peak down the other side of that slope as we kind of pass that threshold. So yeah, we think that's probably something to do with like buffering, basically. Yeah, good question.
Kind of in combination of this and your the second part of your work where you talked about mass and body size, would it be safe to assume, or at least hypothesize, that maybe larger birds might be more sensitive to some of this phenotypical mismatch?
Yeah, that's a good question. Do we see differential impacts on large versus small birds from a phenological perspective? I mean, I guess we'd expect large birds to need more food, even just to feed themselves rather than their young. And if they're also being heat stressed, yeah, I think there's something there. Honestly, I haven't looked at it, but I think you definitely might expect that for sure. And we are doing some work now to try to link with some other folks. Just talk about it at AOS actually. Linking morphological change to demographics. Essentially, yeah, in hotter years are larger birds. Do we see larger birds, and what does that mean for the demographics of the population in terms of population growth rate and breeding success? And we do see a link there. So yeah, but more to do for sure. One question online actually. Okay, question is, what source do you use to determine priors in your bird hierarchical Bayesian models? And do you get those from the literature and determine those yourself? Priors, I am an advocate of setting weakly informative priors. Typically in ecology or in Bayesian modeling, we would set these super diffuse priors where any parameter is possible. That doesn't make any sense. Try to use your field specific, your expertise, your domain knowledge to set those priors informatively, but not so tight as necessarily, hey, we're pulling it out of this paper and really constraining those bounds. And ultimately, at the end of the day, you can look at the overlap between your prior and posterior distributions and see how much your data really impacted that posterior as opposed to your prior, if that answers your question online. Cool, thanks so much for having me, really appreciate it.
Yeah, I would say.
The basics of models in general. Well, that was me. I didn't take any Bayesian. Well, I took a little bit, but it wasn't like in the theories. Yeah. In my. Yeah.